Good morning and welcome to our 5th annual Whitehead Symposium. My name is David Page. I'm the director of Whitehead Institute. And as I fond of saying now, New York City, though we're based in Cambridge, Mass., New York City and in particular the New York Yacht Club are our home away from home. And we've traveled from Boston. We've come here mostly to enjoy the weather, but in addition, it's wonderful to see so many familiar and soon-to-be familiar faces, and I'm deeply grateful that you've chosen to spend the morning with us. Our Board of Associates members generously donate to make our ongoing research and events like this possible. So what I want to do in a few minutes here is convey the essence of what makes Whitehead Institute an extraordinary place for biomedical research. It's a place where the world's most ambitious and curious scientific minds have the freedom to ask and answer the most important questions in biology, questions that will undoubtedly change the way that medicine is practiced and with enormous effects for human health. So Whitehead Institute was the brainchild of Jack Whitehead, who is pictured here together with founding director and Nobel laureate David Baltimore. Jack was an inventor by trade and by nature, and he wanted to create a new model for life sciences research. And Jack's founding vision was quite bold, and the experiment was grand. The idea was to amass a peerless concentration of scientific talent under one roof, here being constructed, and catalyze it with the resources and freedom to blaze new trails. And the results have been profound. Countless breakthroughs from our research and labs have transformed our fundamental understandings of biology and disease, but the full measure of Whitehead Institute's impact extends beyond discovery. So each Whitehead laboratory makes seminal discoveries and shapes new generations of scientific leaders, while Whitehead scientists create vital intellectual capital in the form of companies, technologies, and strategies for leading public and private scientific institutions. So today's Whitehead laboratories are led by 17 distinguished scientists, 17 distinguished faculty who do their research here, and also hold MIT faculty appointments. In addition, 4 Whitehead Fellows. These are a special group of young investigators who come to us right after graduate school or medical school or both, and they're given the opportunity to bypass the postdoctoral phase of training and immediately start making mistakes on their own. For any of you who have started new businesses or been entrepreneurs, you know that really success is about making mistakes. The Fellows Program is about making mistakes as early as possible. And that is, well, we'd actually like to say, creating an independent research agenda. I'm looking at Sebastian. He knows what I'm talking about. So this morning, you're going to hear directly from 2 of our current Whitehead Fellows. And then, in addition to the faculty and the Whitehead Fellows, we have over 300 trainees who come to us as undergraduates, as graduate students earning PhDs, and then also as MDs or postdoctoral fellows. So we have a very large cadre of exceptional trainees. And these scientists together are quite literally doing the research that will change the way that diseases are treated for you and your family. Now, of our current 17 faculty, 3 have been awarded the National Medal of Science. This is bestowed by the President of the United States. It is our nation's highest scientific honor. One of those 3 you'll be hearing from today, Rudolf Janisch. In addition, 5 of our faculty are Howard Hughes Medical Institute investigators, and as of Monday, 11 members of our faculty are members of the National Academy of Sciences. And I'll throw in one little additional statistic. You generally don't get elected to the National Academy of Sciences. It's kind of, it's kind of like the Hall of Fame sort of thing. You generally don't get elected to that until you're in your late 40s. Well, of our faculty under the age of 42, 11 of 12 are members of the National Academy of Sciences. We have 5 faculty who are under the age of 42. So these are ratios without equal in the scientific community. We've had impact elsewhere, in the private sector. Here is a list of some of the public and private companies that were funded and founded, were founded with the help of Whitehead faculty and Whitehead discoveries. And I'll now touch on just a few of the therapies that had their origins, that had their roots in fundamental research conducted at Whitehead Institute. Herceptin for breast cancer, Gleevec for leukemia, Epogen for anemia, tafamidis for familial amyloid polyneuropathy, and Linzess for irritable bowel syndrome. So Whitehead is continuing its legacy of discovery and innovation as exemplified by its next generation of scientists. I'll come back to this. Do you like the idea of being the next generation of scientists at Whitehead?
At my age, I love it.
You love it. We actually are doing some incredible work on regeneration. So we're continuing our legacy of discovery and innovation exemplified by the next generation of scientists. You're going to hear from Sebastian Lourido and Silvia Rouskin in just a bit. But first, I do have a special tribute, and that leads me to the next slide. So, Letty and Tom. We at Whitehead Institute would not be the same without the two of you. Today, we're delighted to celebrate, in a small way, your many contributions to our work. You've long been among Whitehead's most faithful friends. You have introduced Whitehead Institute to a vast, supportive range of colleagues and contacts, a bunch of them here in the room this morning. Letty, you have often single-handedly rallied many to attend our presentations and to find those who would identify with our research and with our mission. Some of you in the room have experienced trying to say no to Letty. You just don't do it. And so the two of you have given generously of your time and of your hospitality, plus you've sponsored our events at key venues in New York City, like this one, repeatedly. And we're enormously grateful for your tireless efforts in introducing Whitehead Institute to meaningful contributors and to new generations of science enthusiasts. So your steadfast belief in our approach to fundamental research has helped us to remain true to our mission and to execute that mission. And so I now have the distinct pleasure of presenting the two of you, if you could come forward, please. I have the distinct pleasure of presenting you with a token of our deep appreciation. We have for Letty and Tom a special, let me see if I can even pick this up.
Oh my God.
Yeah, let me see.
Oh my, it's gorgeous.
Let's see. We have for Letty and Tom this special Simon Pierce Celebration Bowl that is engraved with the names of Letty and Thomas, and that is also engraved with the signatures of all of Whitehead Institute's scientists. So this is a small symbol, Letty, of our appreciation to you and to Tom. And do I get to give you a hug as well?
Absolutely.
While,
With that
okay. All right. Thank you.
Thank you.
All right.
Thank you.
Thank you. Letty, would you like to take this?
Tom and I are deeply, deeply honored. When Tom and I first heard about the Human Genome Project at Whitehead Institute and its potentially stunning impact on the world of health, he and I were instantly hooked. From then on, Whitehead Institute became my project and my passion. I recruited from our circle of friends, from neighbors, even doctors, to participate and volunteer, and I'm happy to see many of you here today. I also want to take this moment to thank the many others who have worked so diligently on behalf of the Whitehead Institute. It is such a pleasure to know you and to spend time with you. And those of you whom I've not met, I look forward to doing so later today or at future Board of Associate events. Volunteering is a team effort, lifelong learning and support, and it's not a solo mission. In quiet and powerful ways, the remarkable work of Whitehead Institute touches our lives and will continue to benefit our children and our grandchildren. But it's more than that. The researchers at Whitehead, the scientists, are also training the next generation of researchers and scientific leaders whose contributions will be felt throughout the world for generations to come. I would like to thank my family and friends for their support and encouragement as we work together to help Whitehead Institute realize its vast potential. Their encouragement means everything. Tom and I could not be more grateful and more proud to be so closely associated with a group such as Whitehead. And thank you all for coming, particularly in this horrible weather. And I hope you enjoy the day. Thank you.
Yeah, I think we can't say enough there, and I'm pleased that we could share a small symbol of our affection for Letty and Tom, how you've supported us in the past and how we'll grow together in the future. So thank you. I now have the pleasure of introducing our two Whitehead Fellows who are with us this morning. We're starting our presentation this morning with an overview of the Fellows Program, which is a unique, high-risk, high-reward feature of Whitehead Institute. As I said, it's about making mistakes early. The program has evolved, and we want to share the success of some of our fellows. I'll just mention two here. Peter Kim was one of the early Whitehead Fellows. He then went on eventually to serve as president of Merck Research Laboratories. Another more recent fellow, Kate Rubens, is now a NASA astronaut. I think we'll be visiting the space station in the not-distant future. And yes, even I was once a Whitehead Fellow. In fact, I was the first fellow at Whitehead Institute. So it gives me special pleasure to introduce two of our fellows, Sebastian Lourido and Silvia Rouskin. They will share their research, and then I'm going to have the opportunity to interview them in front of you. Ask a few questions. You'll have a chance to ask, you'll have the opportunity to ask some questions as well. So now, let's first hear from Sebastian Lourido. Sebastian.
All right, it's really my great pleasure to be able to share some of the work that's been coming out of our lab and some of the vision that we have for what we can do moving forward. I'd like to start this topic by showing you this map. It's one of the earliest nautical maps, and I thought it would be really thematic to the room, that shows a path from Europe to America. And I think it embodies the real sense of discovery and exploration that has been my experience as a Whitehead Fellow over the past few years. Maps, in a more personal note, remind me of how infectious diseases have really shaped human history, have set limitations on particular human populations, and have really changed the way that we interact with the world. One of these infectious diseases, and the one that my lab studies, is called Toxoplasma. It infects nearly a quarter of the world's population. The rate is slightly lower in the US, but 1 in 10 of you are probably infected right now. The good news is that it's typically asymptomatic, and most of you will have no issues. But with such high numbers of patients, in fact, many individuals over the course of their life who become immune compromised will succumb to Toxoplasma infections. And in fact, in places of high prevalence, like Latin America, where I'm from, infection can lead to ocular disease, and over the course of a lifetime can cause blindness. In the lab, we can look at this parasite. It's crescent-shaped, and we can see how it interacts with human cells, shown here as that kind of gray blob. These parasites can move and, in fact, invade the human cells and replicate inside of them, eventually leading to the death of those cells. Many people ask me why we study Toxoplasma, and I've already given you one of those reasons. It's an important human pathogen. But moreover, it serves as a beautiful cell biological model to understand many other important and neglected human pathogens, such as malaria, which all of you know as a scourge of Africa and Southeast Asia, and Cryptosporidium, which is the second leading cause of infant diarrhea, which accounts for 10% of global infant mortality. So many of the related pathogens, the ones that we can learn about through studying Toxoplasma, many of those place tremendous medical and economic burden on some of the least privileged populations on the planet. And the final reason we study Toxoplasma is because it has unique and fascinating biology. The things that Toxoplasma can do to human cells and its ability to persist within our bodies for decades are interesting biological functions that we would love to adapt to medicines, to vaccines, and to many other developments. So these are the 3 dimensions in which we try to think about our research and in which we hope to have an impact moving forward. So we've gotten really good at taking an organism like Toxoplasma and decoding the information in its genome, in its DNA. And that has been in part through tremendous efforts of people like David Page and a former Whitehead Fellow, Eric Lander, who've gotten really good at reading out that sequence and telling us what the sequence of that genetic information is. We also have different experiments and methods to break up that information, which in Toxoplasma accounts for 60 million letters. We can break it up to functional fragments we call genes. There are 8,000 genes in the Toxoplasma genome. So in a way, being able to decode that genome is akin to sending a rocket into outer space and taking a snapshot of the whole globe, right? We can see where the land masses are, we can see where the oceans are, but we still don't know a lot about what the specifics of what those different places in the genome actually do. Which is why when I entered this field and started studying Toxoplasma, I had to kind of take a wild guess and say, I'm going to sail in this direction, I'm going to go to this island, and I'm going to figure out whether it matters for Toxoplasma biology. And I spent 6 years studying 2 or 3 of the 8,000 genes in the genome. This is no way to really move forward, which is why I was so glad to be able to adapt some of the latest technology available for genome engineering in the form of CRISPR. many of you might be familiar with the impact that CRISPR has had on biology. It has many different purposes, and it has been co-opted for many different functions. I'd like to leave in your head like one kind of specific lesson, which is that what makes this so remarkable is that this one yellow protein, which we can introduce into different organisms, can act as molecular scissors that we can target to a specific location in the genome. And in our case, in parasites, we can delete a specific gene. This is done by being able to target with a set of instructions given here in red, a particular sequence, a particular address in that genome shown here in blue. And what we can do is, with high efficiency, we can delete the gene of interest. And because it is so efficient, we can actually take a population of parasites and a large set of instructions covering the entire parasite genome. And we can generate a diverse population of parasites, each one deleted in one of those particular genes. We don't need to choose one location. We can now examine the entire set of 8,000 genes. And what that allows us to do is to then sample what this population can do as they invade human cells and grow over time. And then we can look at the end and say, oh, the gene targeted by the red instructions, that one seems really important because those parasites did not do well. And through this experiment, multiplied by all of the 8,000 genes in the genome, we can derive a certain measure of how important a given gene is to the parasite's ability to infect and replicate inside of human cells. And what I'm showing you here is the diagram of the 14 chromosomes. This is the entire Toxoplasma genome, and each one of these bars is one of those genes. And today I can tell you which one's important and which one's dispensable. I'll concentrate from this trove of information that we've gathered on a single story, and that is a gene in this position that was really unremarkable. We would have never studied this gene were it not for this indication that it was so crucial to the parasite's ability to infect human cells. And today I can tell you that by turning off that gene, we keep the parasites from actually infecting the cells. You can see them trying and not able to enter the human cells that they're trying to parasitize. And motivated by these results, our collaborators have done the same experiment in malaria and shown that this unremarkable gene, one of 8,000, is equally important for malaria's ability to replicate within red blood cells. And this is one of the ways in which studying these basic parasitic processes in Toxoplasma can lead to discoveries not only in that human pathogen, but in the whole vast family of organisms that parasitize humans. And in that way, these genetic experiments that I've outlined this morning are akin to having a live snapshot of the globe and being able to really tell what the populated areas. And what it can do is really broaden our horizons for therapeutic interventions against the scourge of humanity. And with that, I'd like to thank the people in the lab who've really done the work. Most of this was done by trainees in my laboratory named here. And one of the really remarkable opportunities that the Whitehead Fellows Program provides is that of building a team and that of gathering the experience to really bring diverse people together to tackle something that perhaps didn't look promising initially, but that has the potential to really change the way we view these diseases. I'm eternally grateful for the opportunity that the Whitehead has provided, and to many of you who support these efforts. And with that, I'll invite Silvia Rouskin to the stage.
Hi, it's a really great pleasure to be here. Thank you all for coming so I can share my work with you at the Whitehead on finding the second dimension of genetic information. So living organisms are made of just 4 types of molecules. And it's really amazing how this great diversity and complexity of life comes just out of 4 types of molecules. And you may be more intimately familiar with those 3 as common constituents of foods, some of which you enjoyed this morning. But it turns out that the function of carbohydrates and lipids in the body is mainly energy storage. And they're relatively simple molecules. But nucleic acids and proteins, and nucleic acids can be DNA and RNA, are much more complex, and they're essential for life. And historically, we view nucleic acids as the molecules that carry the genetic code. And the genetic code is this instruction manual that specifies proteins. And we view proteins as the molecular machines of the cells, the molecules that do everything that the cell needs to get done. And proteins can make carbohydrates and lipids and do the work that the cell needs. And if we go a step further, conventionally, we think of DNA as the molecule that stores the genetic code, and RNA as the molecule that copies the genetic code and then passes it to make protein, which do the work for the cell. And so what is the genetic code? In the first dimension, it's simply the instructions of how to make a particular machine, a particular protein. And depending on what the cellular needs are at the time, maybe the cell needs to get rid of trash or drill a hole or just needs coffee, the genetic code carries the instructions of which machine should be made. But what specifies the amount? How much of this machine should be made? For example, I don't need 50 coffee machines. If I had 50 coffee machines, that would be a burden to me. I wouldn't know what to do with them. I only need 2. I need just 2 coffee machines, and they have to be at a very specific location. One of them has to be at home. The other one has to be at work. If they were both at work, I just would never go home. And so, similarly, that's equally important for the cell, that it makes the right amount of protein and in the right location. And so what specifies that? Where is the information coming from? It turns out that when RNA copies DNA, it carries a second dimension of the genetic code. And you might ask how. And to understand this, we need to go deeper into what RNA and DNA are and what are the differences between them. RNA and DNA are polymers, meaning poly meaning many, so they're many mers. Or in other words, they're chains of building blocks. And the building blocks are just 4 types. And they're almost exactly identical between RNA and DNA. And when those building blocks come together, they make RNA or DNA. And really the only difference, it's pretty amazing, between RNA and DNA is a single oxygen atom in the building block of those molecules. And in fact, this is where the name comes from. DNA is called deoxy, meaning without oxygen, ribonucleic acid. And RNA is just ribonucleic acid. And the single oxygen atom gives RNA very different properties than DNA. It makes it a lot more reactive and a lot more flexible. And so unlike DNA, which is constrained to just one type of shape, it's always a double helix inside the cell. RNA can form many different complex shapes inside cells. And I really like the analogy with a toy that my daughter plays with called mag formers. And basically, you can build a huge variety of different shapes, and you can immediately appreciate how they have different properties. But all those shapes come from just two types of building blocks, in this case, triangles and squares. And it's all about the way you, the order in which you put them together that gives the shape, the final shape, very different properties. And it's the exact same thing that RNA molecules do, but they just use 4 building blocks instead of 2. We now know that RNA carries a second dimension of the genetic code in the form of RNA shape. So the first dimension, just the linear string of those letters, those bases, specify the identity of a protein, like that this would be a coffee machine. But the second dimension, the shape that the RNA molecule forms, can specify, among many other things, the amount of protein that's made and the specific location that gets made in the cell. And here I've actually taken a figure from our paper to show you that this is the current state of the field. This is how much we know about RNA shapes. But you might ask, since RNA shape is so important, how do we know what the RNA shape is? How can we tell? There are a few ways you can imagine we could figure out what the RNA shape is. A popular way has been, let's make the RNA in a test tube and try to take a molecular picture. And people have tried this. But the issue is that RNA is not rigid. And also there are thousands of different RNAs in the cell. And importantly, it turns out that the RNA shape is actually very sensitive to the environment. And so it's not the same in a cell as it would be in a test tube. So this doesn't work very well for RNA. So what I chose to do instead is to infer the RNA shape through some kind of test. And for this, I chose this chemical called DMS. And the reason why I like DMS is because it's extremely reactive, and it works in a variety of different systems, different cells, as well as organs. And you can look at the RNA shape. And the basic approach is that we take this chemical, we react it with the RNA shape, and then we detect the reaction. And based on that, we figure out what the shape was. And what I did during grad school is to develop a method to determine the shape of all RNA molecules in the cell at the same time. And to explain this better, the basic principle of this, if we take a simple example of 2 simple shapes, one molecule here is very folded, and this one is kind of, so this one is kind of like a hairball folded up, and this one is floppy, like say spaghetti. If we now throw that chemical, let's say a green chemical, and let it react, in the case of the folded shape, it's only going to react with very specific places on the outside of this molecule. But in the case of the open kind of spaghetti shape, it's going to react everywhere. And this is exactly analogous to throwing paint at a person who is in a crouched position, kind of like folded up. What will happen is that only his back is going to get painted in green, but his front is going to be protected. And then when this person comes out, we can tell that he was folded up at the time where we threw the paint. But if the person was open, then he is going to get completely drenched in paint. And in fact, this is the exact principle that I use to determine RNA shapes, only with much more precision. So the methods improvement. So since I've been to the Whitehead, before I came to the Whitehead, we could see the shapes, but they were pretty fuzzy. We could kind of, it's like a fuzzy image that we can tell, maybe this is a face. This is probably a face. Now, since the last year that I've been through the Whitehead, it's actually the method has gotten a lot better, and we can really begin to tell features of this face. Those are eyes, and this is a nose. And the reason why this is so important to tell those features is because those features are what can tell us a lot about what this shape could be doing. If we know this is an eye, that we know it's likely being used for seeing. And of course, the ultimate goal is to get a very nice detailed picture of what the RNA shape is, and that will give us the most information. But we still have, since there's so much unknown, there's so much we can work with having this. And the current lab project is, I'm going to just briefly mention three. One is on RNA localization during development. And here I'm showing images. This is a single cell. In each of those, there's a single cell. And a different RNA molecule is labeled in blue. And what you can appreciate is that every RNA molecule that's labeled here is very specifically localized within the cell. And it's extremely beautiful patterns of localization. But they're not only just beautiful, they're actually very important during development. And this localization sets up gradients that are later going to be used to define which part is going to become a head versus a tail. And so it turns out that the RNA shape is crucial for this localization. The other project we're working on is on HIV, which actually has an RNA genome like many other viruses. Instead of DNA, it codes all this information as RNA. And we actually think that the shape of the RNA could be a key to finally having a cure for HIV, which right now is incurable, and people have to take drugs for their whole life if they're infected by HIV. And finally, a project that we're starting on right now is to figure out what happens to cancer cells. How does the RNA shape changes when the cell becomes cancerous? And lastly, I just wanted to leave you with some food for thought. Our genome has about 3 billion bases, these letters, building blocks of DNA. And it's only a very small portion, just 1.5%, that codes for this protein machines that we're pretty familiar with. But an estimated 10%, a much larger percent, gets converted into RNA. And we now know that RNA can also function as a machine. But RNA machines are completely mysterious to us. And we know almost nothing about them. But you can imagine how knowing their shape will tell us a lot about what they could be doing inside the cell. So with this, I'd like to thank the people in my lab, as well as my collaborators, and a lot of people in the Whitehead. It's really an amazing place to be, and thank you all for coming and listening.
So what we're going to have a chance to do now, I get to interview these two folks. So Sylvie, do you want to take the middle seat over here? Sebastian, you want to go around on the other side? I just want to take a few minutes here we have the chance with Sylvie and Sebastian to learn a little bit more about them and about the Fellows Program. So I don't know if you were really prepared for this, but I think it'd be interesting to hear a little bit more about the two of you personally. So Sebastian and Sylvie, could you just give us your life history in a nutshell? Something. Anyway, Sebastian, take it away. Yeah.
So I was born and raised in Cali, Colombia. I guess that's a good place to start. My mother is a geneticist, actually, a medical geneticist. And so that, I think, kind of early on triggered my interest in science. I went to undergrad at Tulane University in New Orleans, I then spent a couple of years in Berlin working at the Max Planck Institute for Infection Biology, which is where I caught the bug. Then I did my my doctoral degree at Washington University in St. Louis. And since November of 2012, I've been a fellow at the Whitehead.
Okay, all right. So you heard that. So it started at Cali, Columbia, Tulane, Berlin, St. Louis, Whitehead. Okay, all right. Sylvie, take it away.
All right, so I was born in Bulgaria, and my parents are actually both musicians. But I was always really interested in chemistry, and I came to the States when I was 15. I went to Idaho as an exchange student.
You came to the States when you were 15 from Bulgaria.
Yeah.
And where'd you go?
Idaho.
Idaho. Right.
It
okay.
was it was an exchange student program, and that's where people were willing to take me. But I stayed there just for a year, and then I went off to college in Florida, Florida Institute of Technology.
When you were 16.
When I was 16, and then I went to University of California, San Francisco. Which was really great because it's an incredible school in
Did you go there as a student?
No. What happened? What happened is So I graduated college, and I was an international student, and I applied to the top 5 schools in the country, which was Harvard, MIT, UCSF, Berkeley. Stanford, and I got into none of those schools because it was international, and I was coming from this small school in Florida, FIT, and so I didn't want to go to graduate school anywhere else, and I decided I'm going to go and work for someone who I admire. In this case, I specifically stalked a few professors that I really admired, and I was able to get through one of them, and he took a chance on me. He had no idea who I was, but he saw something, and he said, okay, you can work in my lab. And so I worked in his lab for a couple of years, after which I applied to schools again, and then I was accepted everywhere. So that was a very good ending story, happy ending story. And so then I decided to stay at UCSF for my graduate school, and after that I came to the Whitehead.
All right, so there you had it. So Sylvie didn't make it entirely clear. So as she said, she grew up in Bulgaria. Her parents were rock musicians in Bulgaria. Then she somehow made it to, was it Idaho Falls?
Idaho.
A year in Idaho Falls for high school. Florida Institute of Technology, manages to get her foot inside the door in a very exciting lab at UCSF, becomes a grad student, comes to Whitehead. So these are not, I think you're hearing the stories of people who did not necessarily have the road paved for them, right? And those, so anyway, so to each of you, I'd throw out the question then of, why the Whitehead Fellows Program for you? Surely, what were the alternatives? Why did you go with the Whitehead Fellows Program? Sebastian, take it away, or Sylvie, either, you decide who goes first, right?
Hello? Can you hear me? Okay.
Put it up close. Up,
Okay.
yeah.
For me, David, there are actually no alternatives. I only applied to the Whitehead.
I think this is a recurrent theme in your life. You say you have no plan B.
There's no plan B. So I've heard about the Whitehead. I knew about their mission and their vision, and the belief that nothing is really impossible. And I shared that same belief and that same vision, and I thought the Whitehead's got to be the perfect place for me, and turns out it is.
All right, all right. we'll increase your salary slightly now based on those comments. Thank you. Right.
I have the same response. No, I'm kidding. So I also didn't apply very widely because there really aren't very many places like the Whitehead. Right?
What's
The
distinct?
Whitehead
What is
Fellows
this program anyway? Yeah.
So the Whitehead Fellows Program allows you to start your own lab right after your terminal degree, whatever that may be, a PhD, an MD, an MD PhD. And that's really a very unique bet that an institution will place on an individual that hasn't proven themselves as an independent scientist. So you have to take people like David and the rest of the faculty at the Whitehead who have a keen eye and a sense for who these individuals that might succeed pursuing their own research and their own ideas might be. And there aren't a lot of places that are willing to place what is ultimately financially a big bet on individuals like ourselves. And so looking at what the most prestigious programs for that kind of challenge are, there's probably a handful in the US and very few where there was kind of the scientific affinity between what I wanted to do, where I wanted my science to go, and what the faculty were interested in. So I think that really drew me to the Whitehead.
So another question I got, I'll throw out to the two of you. So, and I know this from firsthand experience when I was a Whitehead Fellow myself. The Whitehead Fellows program provides you with a lot of freedom, right? You're not, I mean, when I showed up at the Whitehead Institute a bunch of years ago, I thought vaguely I was actually going to be sort of like a postdoc in Rudolph's lab, to be honest, right? And Rudolph and I have some disputes about this, but I actually arrived before Rudolph did, and I realized I was on my own. There's a lot of freedom. So I'd throw back to you, how have you dealt with the freedom? Does freedom feel good? Does it feel a little risky? How does the freedom that comes with being a Whitehead Fellow feel? How's that worked out for you?
You can go. Yeah. I mean, Silvia and I were talking yesterday about how we both started meditating after we became Whitehead Fellows. I think the challenges inherent to running your own program and developing your own ideas, I think the comparison to starting your own business or entrepreneurship, I think, is perfect because all of a sudden you're in charge for communicating the research, which we're doing today, having a lot of the ideas, recruiting the right people, securing the funding for some of these ideas, which in biomedical sciences don't come cheaply. And so I think all of those challenges are things that as a student I had very little experience with, and that very quickly we had to kind of put together and make those mistakes and really sometimes change course dramatically depending on what the opportunities were and what the failures were telling us about the biological problem. So I think in one dimension, what I presented today with CRISPR, I could have never have planned for, and it wasn't until I came to the Whitehead and I was hearing from people like Rudolph and David Sabatini, who were pioneering these technologies at the Whitehead, that I thought, I think this is something that can really lead to a breakthrough in what I'm investigating in, and I'm going to now devote a substantial amount of my resources to pursuing that avenue. I could have never really thought about that initially. The technology wasn't available, and it would have been a dream to target all 8,000 genes or whatever. So being able to change course and having the supportive network to learn from each other have been two ways in which that freedom has really benefited my research and my career.
So as we pass the mic to Sylvia, I just want to say, Sebastian, you've been a fellow for what, 3.5 years. Sylvie's been here a shorter period of time, so she might have a different take on how this whole thing plays out. Sylvie, tell us about freedom.
So my experience has been, you know, I was a graduate student before, and then suddenly I'm starting my own lab. And I now feel like I have this great power. But of course, you know, with great power carries with itself great responsibility. And I feel this responsibility on a daily basis. I know there is this great expectation of me, and but I know, like myself, like everybody else at the Whitehead, we're all overachievers. And so it's not enough to live up to a great expectation. You have to surpass this expectation. And so, you know, that tends to make us put a lot of pressure on ourselves. But I think as long as we can handle this pressure, I think this pressure can be very good for us and really makes us go the extra distance and not just be pretty good at what we do, but really become the best at what we're doing. And so I think it's really amazing, and I'm really, really grateful to be a part of it.
Great. So, and actually, Sylvia, I can identify with that pressure myself. I think it'll probably fade over the next year or two. I think, based on my own experience, you sort of get used to it. I can say it's really funny. I mean, I came in, both of you at least had a PhD. So I came into the program, I'd been a medical student, but I hadn't even done a PhD. And I remember I was keenly aware of how I would be looking over, you know, I'd be looking in the next door lab at the people who actually had PhDs and were working as postdocs and saying to myself, why am I the Whitehead Fellow with all this independence and freedom? Or as Sylvie just said, the power. But I think you eventually learn, as Sebastian said, that actually as a Whitehead Fellow, you're a scientific entrepreneur, right? And I suspect there's some entrepreneurs in the room. You know that being an entrepreneur is about screwing up and picking yourself back up and doing it over again. And so let's see. I just want to see if there's a burning question or two from the audience that you would like to put to either or both of our fellows. There's one there. Let me see if is there a mic that you can take? Yeah, right. There's right here, right there is the question. Why don't you just announce your name and then your question?
Sure, Evelyn Rodstein. Congratulations on the great work you do. What kind of collaborative peer mechanisms do you have where you learn from each other, even though you're doing independent research?
Yeah, it actually kind of comes natural, a lot of collaborative. First, because everybody at the Whitehead is extremely friendly. They're very kind and very generous with their time, so you can walk into anybody's office and talk to them. So it's the environment is very collaborative, inducive in that sense, and also because I mean I certainly have a lot of collaborations, not just at the Whitehead but also at Harvard and MIT. So I think it's just the environment is extremely well set up for that.
Yeah, we were just talking earlier about a weekly event that we have at the Whitehead where sorry.
That's right.
It's not using microphone. Every Thursday, the faculty and fellows get together, just us, for lunch. And on these occasions, one of us will present our work to our peers and get feedback. And I mean, from my position as a fellow and someone who's just kind of starting out on my independent career, I can't express how impressive and gratifying it is to be able to share a room and present my work and work through my ideas with, as you heard earlier, 11 members of the National Academy of Sciences. It's in a very intimate setting with just 19 of us. It's remarkable to be able to kind of workshop and see what tools are available. And I think that is very much to the spirit of the institution and something that is really remarkable in an academic setting where I think a lot of people are so busy and so divided and so segregated. The Whitehead has been able to generate this space that really embodies the culture of the institution to bring together people who are working on stem cells with people who are working on sex genes, with people who are working on parasitology, and have us all attentively try to tackle the most important challenges that are being identified by each one of our curiosities. And that's a really special place that ends up getting at that. Yeah.
All right, thank you. It's a great question. And is there one? Yes, Jim. State your name and your question, please. Yeah.
Being a fellow is exciting, and having the independence that you have is terrific. Where do you see your activities 5 years from now? Not your scientific activities, but will you be hopefully still at the Whitehead? Will you go to another research institute, to the industry, and so forth?
I'm definitely going to still be running a lab. And I think the Whitehead is an amazing place. It's extremely difficult to stay in the Whitehead because, it's part of MIT. It's very competitive. But for sure, this really prepares us to be able to run a lab for the rest of our lives. And I think it's extremely exciting. And so we will try, all of us, to stay at the Whitehead. If not, we'll go through, Harvard or some other place.
So I'm an empiricist, right? I like experiments. And if you think of the Whitehead Fellows Program as, what is it now, a 30-year experiment or so?
32-year experiment, yeah.
There have been approximately 34 Whitehead Fellows. And when you look at the roster, there's one that went to NASA, so that's a little bit hard to predict. But most of them are still leading, really pioneering research programs, either in academia or in industry. Most of us end up staying in academia because I think there's something very special about the freedom that academic research positions give us. And that's something that's very compatible with the type of people who end up being Whitehead Fellows, right? Who are driven to pursue their own goals and their own ideas. But a number of us have become leaders of research in industry, and that's certainly another possibility. So I'm, of course, with all of the anxiety that comes with planning one's future, I am somehow heartened by the track record that this program has.
All right. I think we should give it up for Sylvie and for Sebastian for taking our questions in these great presentations. Thank you both. Thank you, Sylvie. Thank you, Sebastian.